So now we're going a little bit out of order in our textbook. We are going to do chapter 12, talking about neurons, before we do chapter 10, which talks about muscles. And hopefully it'll make sense to you why many of us choose to teach this chapter first, because it is going to have to do with how our muscles contract. So we're going to start talking about the nervous system. When we talk about the nervous system, we are talking about the brain and spinal cord, the receptors of our sense organs, such as our eyes and our ears, and then the nerves that leave our central nervous system, that leave the brain and spinal cord and connect to other systems. And the nervous system accounts for about 3% of our body's weight. And this image here is basically all the components of our nervous system with the rest of the body taken away. So you can see the brain, the spinal cord, and those are all the peripheral nerves coming off of the spinal cord. So when we talk about our nervous system, we have anatomical divisions and functional divisions. So anatomical meaning where in the body the components are located, and functional meaning their function. So anatomically we have the central nervous system, which includes the brain and spinal cord, and then we have the peripheral nervous system, which is all the nerves that extend into the periphery from there. For functional divisions, we have the central nervous system, the peripheral nervous system, and the enteric nervous system. So let's talk about the central nervous system first. The central nervous system consists of the brain and spinal cord, and it's made up of nervous tissue, supportive connective tissue, and blood vessels. The function of the central nervous system is going to be to process and coordinate sensory data coming from both inside and outside of our body, and to control motor commands, sending information about what our peripheral organs, such as skeletal muscles, should be doing. And then also what we call higher functions, which when we say higher functions, we're referring to things like intelligence, memory, learning, and emotions. The peripheral nervous system is that nervous tissue located outside the central nervous system and outside the enteric nervous system. The function of the peripheral nervous system is to deliver that sensory information that's coming in from outside our body or from our internal organs to the central nervous system, and then to also carry commands coming from the central nervous system out to peripheral tissues. The structures of the peripheral nervous system are our nerves, also known as peripheral nerves. And these nerves are made up of bundles of axons, or nerve fibers, with connective tissue and blood vessels. And we'll look more closely at the structure of a nerve later. So those nerves are going to carry sensory information in, send motor commands out, and we also have cranial nerves that are connected to the brain itself, and spinal nerves that attach to the spinal cord. When we talk about functional divisions, the central nervous system does not have, it's not divided into functional divisions, but the peripheral nervous system is. The functional divisions of the peripheral nervous system include the afferent and efferent divisions. The afferent division is going to carry sensory information to the CNS. It's taking information from receptors in peripheral tissues and organs and bringing them into the central nervous system. These receptors are going to detect changes in the environment or respond to stimuli. The receptors could be neurons, or they can be specialized cells. And we might have a single cell, or we could have a complex sensory organ, such as our eyes or our ears. So the afferent, with an A, arrives at the CNS. And so I have some little memory devices that I use. Afferent arrives at the CNS. So afferent is bringing all that sensory information into our spinal cord and our brain. Another mnemonic that's used is the word same down here. So we say sensory is our afferent, motor is our efferent. So we've got same. And if you think about sensory information is coming in, arriving at our brain. Motor commands are going to be leaving our brain to go to peripheral effectors like our muscles. So that was the afferent division that we just mentioned. Then we also have the efferent division. The efferent division is going to be carrying motor commands from the central nervous system out to muscles, glands, and adipose tissue. So for this one, I say E is for efferent, exiting the CNS and going to our effectors. So when we say effectors, that's the target organ that's responding to these motor commands that are being carried away from the brain and spinal cord. So once again, the afferent division, sensory information coming into our central nervous system. Efferent division is motor commands exiting and traveling to our effectors, which is whatever part of the body is going to be responding to those commands. The efferent division can further be divided into the somatic and autonomic nervous systems. So when we say the somatic nervous system, this is going to be our skeletal muscle contractions. And this includes both voluntary and involuntary movements. Most of our skeletal muscle is going to be voluntary movement. You can think about, oh, I'm gonna, move my hand or turn my head side to side. You can think about that. But there are some involuntary movements, such as reflexes, that are part of the somatic nervous system response. And then we have the autonomic nervous system. The autonomic nervous system is responsible for subconscious actions, things you are not aware of. And it's going to be responsible for contractions of our smooth and cardiac muscle, as well as glandular secretions in adipose tissue. And then the autonomic nervous system can further be divided into the parasympathetic and sympathetic divisions. And these have antagonistic effects in general, meaning they kind of work opposite of each other. The sympathetic system is usually thought of as the fight or flight system and usually has a stimulating effect. For example, if the sympathetic division is activated, your heart rate is going to increase, whereas the parasympathetic division is called the rest and digest branch and usually has a more relaxing effect on our body. And then just a brief note on the enteric nervous system. This is the third functional division of the nervous system, and it includes neurons in the walls of the digestive tract. There are as many or more neurons in the walls of the digestive tract as are in the spinal cord. And they're going to use the same neurotransmitters as the brain. They are going to be responsible for coordinating visceral reflexes, so reflexes of the abdominal viscera, the enteric or digestive system. And they are going to control those visceral reflexes locally without instruction from the central nervous system. So our digestive tract can function without input necessarily from the brain, but it can also be influenced by our autonomic nervous system. So the autonomic nervous system, remember, we said divided into sympathetic and parasympathetic branches. So depending on what's going on with the autonomic nervous system, we can also affect the enteric nervous system. And this is just a little flow chart from your textbook. So we've got the central nervous system, brain and spinal cord, and then the peripheral nervous system, all those nerves outside the spinal cord. We've got the central nervous system is going to be responsible for all that information processing. So it's going to receive these sensory information coming in from that afferent division, and then it's going to send out commands in the efferent division. The sensory information that's coming in could come from special sensory receptors, such as monitoring our special senses, our sight, sound, smell, visceral sensory receptors, monitoring our internal organs, and then somatic sensory receptors, meaning monitoring the surface of our skin, our joints, and our skeletal muscles. So all those receptors are going to transmit information via the afferent division of the peripheral nervous system up to the CNS. The CNS is then going to decide what do we need to do with this information that's coming in, and then send commands out via that efferent division of the peripheral nervous system. And then that efferent division further divided into the somatic nervous system, controlling our skeletal muscles, and the autonomic nervous system, which is going to be controlling that smooth muscle, cardiac muscle, glands, and adipose tissue. And then that autonomic nervous system is further divided into parasympathetic and sympathetic divisions, which we'll talk more about at the end of the semester. Just a quick note about terminology and anatomy in this unit. A lot of times it can be easy to, when we're talking about nerves or neurons in this unit, to confuse the two. So neurons are cells, right? We talked about neurons briefly when we were talking about tissues. So this on the left is a neuron. It's got a cell body, it's got that axon and those dendrites, and it's going to be collecting information via those dendrites, sending it down the axon to our synapses, which are going to communicate with another cell, another neuron, or a muscle cell, etc. So that's our neuron, single cell. When we talk about a nerve, and that's what we're seeing in this image on the far right, when we talk about a nerve is made up of many neurons, as well as other tissues, such as connective tissue and blood vessels. And so you can see in this image in the middle, this structure that's kind of pulled out here is a single neuron. And we have lots of those neurons organized in bundles. So each of these that I've circled is a bundle made up of many neurons. And then those bundles of neurons are further grouped together in a larger bundle with blood vessels and surrounded by a sheath of connective tissue. And this is what a nerve is. So when we're talking about neuron versus nerve, I just want you to have a mental image and not mix the two up. So we talked about nervous tissue already. Nervous tissue contains two kinds of cells. What were the cells? We just talked about one of them in the last slide, right? Take a minute and quiz yourself. Number one in this image is going to be our neurons. So they are responsible for intercellular communication. And then the other cell, number two, is going to be our neuroglia, or glial cells. And these glial cells are essential to survival and functioning of the neuron. So they're a very important part of the nervous tissue. They're going to preserve the structure of the nervous tissue and protect, nourish, clean up, and regulate that nervous tissue. And we're going to go through and talk about the different neuroglia cells. So those neurons are the basic functional unit of our nervous system. They're responsible for communication, processing information, and controlling the rest of our body. They're going to be sending signals and also receiving signals. And they have what's called an excitable plasma membrane, which we'll talk about in much more detail in the next lecture. But what we mean by excitable membrane is that they're able to generate and propagate action potentials. This requires energy, as does secretion of the chemical compounds for sending information, the secretion of neurotransmitters. And so our neurons have mitochondria, which are going to generate this energy. Our neurons have no centrioles. Do you remember what centrioles were involved in back at the very beginning of our semester? They're involved in mitosis, that cell division. So a typical neuron does not have a centriole, which means it can't divide. So if we lose a neuron to injury or disease, it won't be replaced. We do have neural stem cells that are present in adults, but unfortunately they are inactive. So we have stem cells there that could potentially divide and make more neurons, but they are not active in adults. There are some exceptions to this. The olfactory receptors in our nose and neurons in our hippocampus, which is involved in memory storage. We do have some active neural stem cells there. And scientists are currently trying to figure out how to trigger neural stem cell activity, and that would be used to aid in brain injury treatment. If we could figure out how to do that, it would be great. When we look at the structure of the neuron, we have the cell body or soma, and then within the cell body, we have a large nucleus and nucleolus. So our cell body is going to be, this is our cell body. We have a large nucleus and nucleolus. The cytoplasm within the cell body is called the perikaryon. And we've got mitochondria there that produce that energy that we need for generating action potentials, which is how we send our signal. There are dense areas of rough endoplasmic reticulum and ribosomes that synthesize protein in that perikaryon, that cytoplasm. And they are what makes nervous tissue appear gray. You might have heard the terms white matter and gray matter when talking about the nervous system. And so that gray matter, that darkness to the gray matter, comes from these areas of rough endoplasmic reticulum and ribosomes that have that dark appearance. The perikaryon also has a cytoskeleton, which is made up of neurofilaments and neurotubules, similar to tubules within the other cells that we talked about, and neurofibrils, which are bundles of neurofilaments that support those dendrites and support the axon. So we do have some supportive structures within these cells to help maintain their structure. Coming off the cell body, we have what are called dendrites, and that's these little branch structures that we see here. These dendrites are short and highly branched processes that extend from the cell body, and they have a key role in communication between cells. Some of them have little spines, which are little tiny processes on the dendrites, and you can see that in the image down at the bottom here. This is a dendrite, and it's got all these little spines coming off of it. And if you think about what that might do, that structural shape, think about the microvilli and how that increases surface area. This is going to be similar. Those fine processes on the dendrites are going to increase the surface area where we can receive information coming from other neurons. So 80 to 90% of the neuron surface area is actually on the dendrites themselves. So coming off the cell body, we have what's called the axon. That axon is a single long cytoplasmic process, sometimes up to 1 meter in length, and it's going to be responsible for propagating or carrying along electrical signals, which are called action potentials. And we have some other terms that relate to this. We have the axon hillock, which is a thickened region where it attaches the initial segment of the axon to the cell body. So here's our initial segment of the axon, here's our cell body. So the axon hillock is this area where they are connected. That initial segment is the base of the axon, and it contains what's called the trigger zone, which is going to be involved in our action potentials we'll discuss later on. We have a special name for the cytoplasm within the axon as well. Anatomy likes to have special names for everything. So the cytoplasm within the axon is called the axoplasm. And then the plasma membrane on the axon is called the axolemma. We can also have what are called collaterals, which are branches coming off the axon, and that allows one neuron to communicate with multiple other cells. And there's no collaterals on here, but if we had a collateral, we would see these little branches coming off the axon, and those would be collateral branches. And then we have telodendria, which are also known as terminal branches. And it's basically the extension of the end of the axon, branches off. And at the end of these telodendria branches, we have what are called our axon terminals, or sometimes called our synaptic terminals or our synaptic bulbs. And these are the tips of those telodendria, and this is where communication is going to occur with another cell. And we call that area where one cell communicates with another the synapse. And that's why they're called synaptic bulbs or synaptic terminals. So this is another view of, I think this one is taken from your book. So cell body, dendrites, picking up information, so information is going to come in here. Axon, you can have collateral branches coming off of this axon. Telodendria, where that axon is branching out. And then at the end of the telodendria, we have those synaptic bulbs or axon terminals. And these are the site where the neuron is going to communicate with another cell. And so that's called the synapse. We can move materials between the cell body and the axon terminals, so from one end of the axon to the other, via a process called axonal or exoplasmic transport. And these materials move along neurotubules within the axon. And we have special proteins which help move these materials along with the help of ATP coming from mitochondria. So we have proteins called kinesin and dynein that move these materials along the axon. And you can see in this image on the right is kind of a representation of the materials kind of being pulled along those neurotubules by those special proteins with the help of ATP coming from those mitochondria. And we can move materials quickly or slowly. We have slow stream movement, and with slow stream movement, we're just moving a few millimeters per day, whereas fast stream movement can be up to 1,000 millimeters a day, so much quicker. We can also transport things in different directions. We can move things from the cell body to the axon terminal, and this is called anterograde flow, and the kinesin protein is what's going to be responsible for anterograde flow. And then we can also move from the axon terminal towards the cell body, and the dynein is going to be responsible for that movement. So, big takeaway here, we have movement from, use a different color, cell body to axon terminal is anterograde, and then movement in the other direction from the terminal to the cell body is retrograde. And we can have fast or slow movement there. So one good example of retrograde flow is the rabies virus. I obviously am interested in this virus being a veterinarian, but it can affect humans as well. And it is a kind of interesting virus the way it travels. So I'm just going to draw a quick representation of a person here. Bear with my awful drawing skills. I just want to give you the idea of where things are happening here. It's really hard to draw with your finger on a computer. All right, so there's our person. And that person is going to have their central nervous system. So we've got their brain and their spinal cord, right? And then they're going to have a skeletal muscle in their leg. So this is their skeletal muscle. And then we're going to have the peripheral nervous system. We're going to have nerves that come down from the central nervous system and innervate that skeletal muscle. And then we're going to have nerves that go from that skeletal muscle to the central nervous system. I should make this, I don't know how to make this thicker. Oh well. So nerves coming down to and nerves going back. So quiz yourself. When we have these nerves that are coming down to the skeletal muscle and innervating the skeletal muscle, carrying messages from the central nervous system to the skeletal muscle, is that the afferent or efferent branch? It's going to be the efferent branch. And then these nerves that are bringing signals from that skeletal muscle up to the brain is the afferent branch. We can also divide the peripheral nervous system into somatic and autonomic branches. So think to yourself, is this the somatic or the autonomic branch of the peripheral nervous system if we're talking about nerves innervating the skeletal muscle? So that's going to be the somatic branch of the peripheral nervous system. Okay, so what happens with the rabies virus is you have, unfortunately, some little animal who is infected with the virus, and the virus actually does some interesting things. It does affect the brain eventually, and so it makes animals oftentimes more aggressive. It also increases their flow of saliva and makes it difficult for them to swallow. And so what happens is that virus replicates within the salivary glands. And so we have lots of viral particles in the saliva. The animal can't swallow. So we have even more saliva in the mouth. And then the animal becomes aggressive, which is good for the virus because that animal is then going to bite the person. So this is our bite right here. And those viral particles that are in the saliva are going to be injected into that skeletal muscle. So those viral particles enter the skeletal muscle, and then what happens is they travel via retrograde flow up those efferent nerves via retrograde flow in the neurons. So it's going to travel via retrograde flow from neuron to neuron. Remember the nerves are composed of multiple neurons. It's going to travel slowly back up to the central nervous system. And luckily, the flow of the rabies virus is a slow stream flow because once the virus reaches the brain, there really is very little chance of survival, like 99.9% fatality rate. So luckily, the virus is slow stream. It moves very slowly, which gives us time to treat people who may have had a potential bite from a rabid animal. So there's a link here to more information on the rabies virus if you're interested. But rabies virus retrograde flow, slow stream. Our individual neurons can be classified based on their structure as well as their function. So when we look at the structural classification of neurons, they're kind of named by the processes that are coming off the cell body. So we have anaxonic neurons, which means anaxonic without axon. And these neurons they have small processes that are all similar, like dendrites, look like. You can see here the branching appearance. And we find these anaxonic neurons in the brain and the retina of the eye. Then we have bipolar neurons, which means we have two processes coming off of the cell body. So we have 1, 2. And one of those processes is a long process with dendrites on the end, and the other process is an axon with an axon terminal. And those bipolar neurons are fairly rare, and they're found in special sense organs such as the eyes, the nose, and the ears. Then we have unipolar neurons. And with unipolar neurons, it looks here like very similar to the bipolar, like we might have two processes coming off. But if you look closely, I'm just going to draw another representation. We've actually got the cell body here, and then there's one process coming off that then splits into different branches. So really, we have one process coming off of that, one long process coming off of that cell body that then branches. And we find this type of neuron in the peripheral nervous system. And then the neuron that we have discussed the anatomy of and that we'll be talking about in these next units is a multipolar neuron. So make sure you know multipolar neuron. And this neuron has numerous processes coming off of it. It's got that long axon, and then it's got all these dendrites coming off. So it's got multiple processes coming off of it, multipolar. And this is the most common neuron in the central nervous system. And the motor neurons that control our skeletal muscle are multipolar neurons. So that was the structural classification. We can also have functional classifications of neurons. We have sensory, motor, and interneurons. And then these can be further divided into different categories. Anatomists love to divide things and categorize them. So our sensory neurons. The sensory neurons are going to form the afferent division of the peripheral nervous system. Remember, draw our little. Here's our central nervous system. So afferent is going to be bringing information into the central nervous system, arriving at the CNS, and it makes sense that our sensory neurons would be part of that branch because it's our senses, and those senses need to come in and bring information to the brain, to the central nervous system. And so these neurons are a part of the peripheral nervous system as well. So the afferent division of the peripheral nervous system. The cell bodies of these neurons are found in what are called peripheral sensory ganglia. And a ganglia is the term used for a collection of neuron cell bodies in the peripheral nervous system. So oftentimes we'll find cell bodies that are all kind of grouped together in one structure, and those are called ganglia. These neurons are that unipolar neuron, so have one process coming off that then splits into the axon and the dendrites. And those processes are called afferent fibers. And those afferent fibers, those processes extend from sensory receptors in the peripheral body to the central nervous system. So those processes are going to extend from, we'll have, let's see, it looks like in this picture on the right, this is our axon terminal with our synaptic bulbs, and then this would be our dendrites. So remember, dendrites is where we're picking up the information. So information is going to arrive at those dendrites and be carried along those afferent fibers to the central nervous system. So those fibers are going to be traveling from the periphery up to sensory receptors, from the sensory receptors to the central nervous system. We have both somatic sensory neurons, and those somatic sensory neurons are going to monitor the external environment and our position within it. And then we have visceral sensory neurons. And if you remember talking about viscera before when we were talking about the serosa, remember we had parietal and visceral serosa, and the visceral serosa, we said the viscera were the abdominal contents. So the visceral serosa, the visceral sensory neurons are going to monitor those internal organs. They're going to monitor the environment and status of our internal organ systems. So let's talk about the different types of sensory receptors. We have different types of sensory receptors. We have interoceptors. Intero means inside, and they're going to monitor internal systems like our digestive and urinary tract. And I put this picture in here to help you remember it. They're going to sense internal signals such as stretching. In this picture, the bladder is stretching and this individual is sensing that their bladder is full and they need to use the restroom. They can also sense deep pressure and pain. Then we have exteroceptors, and extero is going to be referring to outside. And so the exteroceptors are going to provide information about the external environment, such as the temperature, touch, and pressure. And then we also have proprioceptors. Oh, sorry, I missed. The exteroceptors are also going to be involved in our complex senses like sight, smell, and hearing, which are monitoring things in the external environment as well. Then we have proprioceptors, and proprioceptors, they monitor our position and movement of our skeletal muscles and joints in relation to the world around us. So they kind of sense where our body is and how our body is moving in relation to things around us. So if you closed your eyes, you would still be able to walk across the room or even probably walk up a flight of stairs or up a ladder because your body has an idea of where it is in relation to the environment. If you had a problem with your proprioceptors, your body would not know where it was in relation to the environment, and you would probably end up stumbling or falling. So those were our sensory neurons. Now we're going to talk about our motor neurons. So our motor neurons are going to be the efferent neurons. So remember, we've got our central nervous system again. So our efferent neurons are the neurons that are going to be exiting or leaving the central nervous system and traveling to peripheral effectors. And they travel via efferent fibers. So those processes on those multipolar neurons are going to be called fibers. Those axons are called fibers, and these are efferent fibers because we are exiting the central nervous system. So these are those multipolar neurons that we talked about where we have one long axon and then lots of dendrites coming off the cell body. And we have both somatic and visceral motor neurons. So somatic are going to be sending signals to things like our skeletal muscle to move, and then visceral are going to be sending signals to our internal organs, giving commands on what we need to do there. So somatic motor neurons of the somatic nervous system are going to innervate skeletal muscles, as I said before. Most often this is conscious control, so you think about flexing your bicep or moving your leg and you can do it. You have conscious control, you're aware and telling your body to do that. The cell body of these neurons that make up these nerves is located within the central nervous system. So you can see in this image down here, this is the cell body here located within the spinal cord. And then the axon exits the central nervous system and runs via a peripheral nerve. Remember, our nerve would be made up of many of these neurons, all grouped together. So that those axons of those neurons are going to run via a peripheral nerve to innervate the muscle, which is our effector, at what's called a neuromuscular junction. That the neuromuscular junction is where that neuron is going to touch that skeletal muscle. With our visceral motor neurons, they are part of the autonomic nervous system, and they're going to innervate all the other peripheral effectors other than that skeletal muscle. So we have smooth and cardiac muscle, glands, and adipose tissue. And this division is unconscious control. You do not think about your heart beating. You do not think about your digestive system moving. With these visceral motor neurons, the cell body is going to be in the central nervous system again. Here we've got one in the spinal cord. But then what's going to happen is that axon is going to leave the central nervous system, and it's going to travel to a second set of visceral motor neurons in the that begin in the autonomic ganglia. So remember, we said a ganglia is a group of cell bodies in the peripheral nervous system. So we're going to have our first neuron originating in the central nervous system. Its axon is going to travel out, and it's going to communicate with a second neuron whose cell body is in this autonomic ganglia. And then the axon of that neuron, that second neuron, is going to go and innervate our visceral effector, which is going to be our smooth muscle, our cardiac muscle, our glands, etc. We can, when we're naming neurons, we'll sometimes refer to preganglionic or postganglionic fibers when we're talking about the axons of this neuron. So preganglionic fibers would be the axons of the neuron that's directly leaving the central nervous system. And then postganglionic would be those fibers that are leaving the ganglia to innervate the visceral effectors. So that signal travels from the central nervous system along an axon. Let me erase this real quick. So in this instance, that signal is going to come from the central nervous system, travel along an axon, where that signal is then going to cross the synapse from that axon terminal to a new cell body in that autonomic ganglia. And then the signal is going to travel along that postganglionic axon or fiber to reach its final destination, its final peripheral effector. So our third type of neuron are our interneurons, also known as association neurons. And there are lots of these, most of any neuron, 20 billion of them. Most of them are found in the brain and spinal cord, but we do have some in those autonomic ganglia. Remember, those autonomic ganglia are kind of groupings of cell bodies in the peripheral nervous system. These interneurons are in between sensory and motor neurons, so they're kind of like a little relay center, passing information from the sensory neuron to a motor neuron. They are responsible for integration. So they are bringing sensory information in and deciding where it needs to send and where it needs to go to, and then coordinating motor activity. So they receive that signal from the sensory neuron and determine, does it need to go up the spinal cord? Does it need to go to the brain? Does it need to go out to a motor neuron? And they are also involved, the ones that are within our brain, in higher functions. And when we say higher functions, remember that means things like memory, planning, learning. So those were all our neurons. Now we're going to move on to that other cell in nervous tissue, the neuroglia or glial cells. So the neuroglia we mentioned when we talked about tissue, the first time we talked about tissues, are those supportive cells for the nervous tissue. They're going to support and protect our neurons. They provide a supportive framework for our nervous tissue. Some of them can be phagocytic, and they help regulate the composition of interstitial fluids in the nervous tissue. Remember, interstitial fluid is that fluid outside of the cell. And neuroglia make up about half the volume of our nervous system. We've got lots of different types in the nervous system. There are different types in the central versus the peripheral nervous system, and we're still learning more about these cells because they have been difficult for scientists to study. So the types of neuroglia we find in the central nervous system include astrocytes, ependymal cells, oligodendrocytes, and microglia. And then the peripheral nervous system, we only have two types, satellite and Schwann cells. So let's talk about the ones in the central nervous system first. So astrocytes are going to be large cells. They have a large cell body with lots of processes coming off. And they are the largest and most numerous neuroglia in the central nervous system. They're called astrocytes because if you use your imaginoscope, they have a star-shaped appearance with lots of these thin cytoplasmic processes which end in feet. So in this image here, this guy is our astrocyte. So he's got this big cell body and lots of processes coming off to make him star-shaped, I guess, if you use your imagination. And then his little processes end in these little feet that you can see here. And what these cells do is they help maintain the blood-brain barrier. The blood-brain barrier is a filtering mechanism for capillaries that are carrying blood into the brain or spinal cord. And the blood-brain barrier provides a selectively permeable barrier. Remember, selectively permeable means some things are allowed in, but others are not. So those little feet coming off those processes of the astrocytes, they kind of spread out and form a little blanket around the capillaries that are running through that nervous tissue. And they secrete chemicals which are responsible for that selective permeability. So those chemicals basically tell the body what is going to be allowed into the nervous system and what is not. And the blood-brain barrier can be important in medicine because there are some medications that are able to cross the blood-brain barrier and others that are not. And so if, for example, you had something like a meningitis and it was a bacterial meningitis, and you would need to make sure you selected an antibiotic that would cross the blood-brain barrier if you wanted to treat it effectively. The astrocytes also provide a three-dimensional structural framework for the central nervous system, and they are involved in repairing some damaged nervous tissue in the brain and spinal cord. Unfortunately, their repair capabilities are limited. They cannot restore full function, but they do sometimes form some scar tissue that kind of stabilizes the area and prevents further damage. They're also responsible during embryonic development of the brain for guiding the neuron development, telling those neurons where to go, where to grow to. And they help control the interstitial environment. Remember, interstitial is outside of the cells, by regulating concentrations of things like sodium, potassium, and carbon dioxide. They are also going to be involved in transportation of nutrients, ions, and gases between the capillaries and neurons. And they absorb and recycle some of the neurotransmitters that are found in the nervous system. And they can release chemicals that enhance and suppress communication across our axon terminals. But the big thing to remember for these guys is they are what form our blood-brain barrier. And this is an image from your textbook, and you can see here the astrocyte is this cell here. Here's the cell body, and you can see all the processes coming off that end in these little feet. And you can see here's a capillary running here, and you can see that those feet on those astrocytes basically engulf that capillary, providing that barrier that's going to be selectively permeable. Our ependymal cells form an epithelium that lines the central canal of the spinal cord in the ventricles of the brain. The central canal of the spinal cord is basically a fluid-filled central passageway that passes through the longitudinal axis, so longitudinally right down the center of the spinal cord. As that central canal reaches the brain, the passageway enlarges into chambers within the brain that are called ventricles. And you'll learn more about those in our next unit. So these ependymal cells form basically the lining of this canal and chambers, and they're going to help produce and monitor our cerebrospinal fluid, the fluid that's circulating through those chambers. And some of these ependymal cells actually have cilia. Remember, cilia are those little processes that can move in unison to move fluid. And so in the third ventricle of the brain, these cilia help circulate our cerebrospinal fluid. And so in this image, let me do red. These are our ependymal cells here. And this would be like the central canal or ventricles with the fluid. And you can see the cilia here at the surface of those cells. And you can see these ependymal cells basically form this layer of cells between the nervous tissue in the brain or spinal cord and then that cerebrospinal fluid. And in this image here, your ependymal cells, this one is from your textbook, are these cells right here. So here's our central canal and the spinal cord, and then you can see these cells form a barrier, and these ones have some little cilia as well. Our oligodendrocytes have small cell bodies and fewer processes than the astrocytes. So they're smaller with fewer processes than those astrocytes were. Those processes come into contact with the exposed surfaces of neurons. And when these oligodendrocytes are mature, the plasma membrane at the tip of each of these processes kind of stretches out and expands into this thin pancake, and then that thin pancake of cell membrane wraps around the axon of the neuron, and it forms these concentric layers of plasma membrane, and that's what forms a myelin sheath. You may have heard that term before. So many of these oligodendrocytes are going to cooperate to form myelin sheaths along the axon of a neuron. And in this image, so here is our oligodendrocyte, and you can see it has these processes coming off. So this is actually one process of an oligodendrocyte that had flattened out and wrapped all the way around this axon. And the axon is this blue area in the center. So this is the axon of this neuron here. The axon's running down, and this oligodendrocyte has had these processes which flattened out and then wrapped and wrapped and wrapped around this axon to provide that myelin sheath within the central nervous system. And myelin is important because it acts as an electrical insulator for those axons, and this allows the speed of action potentials, or basically the message transmission down that neuron, to be much faster. And we'll talk more about that when we talk about action potentials in the next lecture. Each oligodendrocyte myelinates segments of several different axons. So this oligodendrocyte that we have here, let me just erase what I just did. This oligodendrocyte that we have here, it has might have processes that have expanded and myelinate this neuron, and we might have another neuron over here. So here's our neuron, this is our axon here. It might have other processes that come over here and then myelinate this axon as well. These myelinated areas of the axon do contain a lot of lipid, and that is what makes our nerves appear white. The amount of lipid within this myelinated sheaths makes the tissue have a white appearance. So you might have heard of white matter and gray matter in the nervous system before, and this is where that white matter comes from, those myelinated axons. And this is just an image from your textbook. So here is our oligodendrocyte. Here's the cell body. Our axons are running here. And then this process of the oligodendrocyte has come off and thinned out and then wrapped itself around and around that axon. And then it's come over here, and the same thing, it's wrapped around and around that axon to myelinate it. A little bit of terminology when we're talking about myelinated axons and their anatomy. An internode is the name given for that myelinated segment of axon. So each of these areas that are myelinated are called internodes. And they're 1 to 2 millimeters long usually. In between the internodes, we have little unmyelinated sections, and those are called the nodes of Ranvier. And these are micrometers small. And if we had a neuron where we had collateral branches coming off of the axon, this is where the collateral branches would exit. Remember, those collateral branches would be little branches leaving the axon to end in different axon terminals to connect with other neurons. So if we had those collaterals, they'd come there. As I mentioned before, sometimes we talk about white matter and gray matter in the central nervous system. So white matter is going to be areas of the central nervous system where we have lots of these myelinated axons, and gray matter is going to be areas where we have unmyelinated axons. We are seeing more the cell bodies of neurons and the dendrites. And then our microglia are our smallest and least numerous neuroglia. These ones are small with lots of fine branch processes, and they migrate through the nervous tissue, acting like a little wandering janitor or police officer. They are phagocytic. Remember, phagocytes are cells that might eat pathogens and debris, so they are going to be responsible for cleaning up cellular debris, waste, and pathogens. And they originate from the same stem cell that produce macrophages and monocytes. Remember, macrophages were our big eater cells. So these cells are going to be kind of cleaning up our nervous tissue. And in this image here, you can see our microglia is this small cell with lots of fine processes. So that was the central nervous system. In the peripheral nervous system, the cell bodies of the neurons in the peripheral nervous system are clustered in what we call ganglia. Remember, we mentioned those before. So those cell bodies, remember our neurons got a cell body, an axon, its telodendria or axon terminals, and then it's got the little dendrites up here. This is a very simplified drawing. So all those cell bodies can be grouped together into ganglia, clusters of cell bodies in the peripheral nervous system. The processes of our neuroglia are going to completely insulate the neuronal cell bodies and most of the axons from their surroundings in the peripheral nervous system. And those neuroglia are going to be called satellite cells and Schwann cells in the peripheral nervous system. So satellite cells are going to surround those ganglia, and they help regulate that interstitial fluid around the neuron, similar to the astrocytes regulating the interstitial fluid in the central nervous system. Our Schwann cells, or neurolemmacytes, are the cell that's going to be responsible for forming myelinated sheaths around the axon in the peripheral nervous system. So we had the oligodendrocytes myelinating axons in the central nervous system. We have Schwann cells myelinating in the peripheral nervous system. So these Schwann cells can either form myelin sheaths, or sometimes they... Grandma's here. Sorry about that. So they will either form myelinated sheaths similar to what we saw in the central nervous system, or they can form these indented folds of plasma membrane that surround peripheral axons. So in this image on the right, this is our Schwann cell, and you can see it's got this little indentation that's surrounding a group of axons. So the neurolemma is the term given for the outside of the Schwann cell that's covering the axon. And that neurolemma is going to shield that axon from the interstitial fluid. So it's just another layer of protection. Those non-myelinating Schwann cells, the ones that I was talking about here on the right-hand side, they're going to enclose segments of several axons that are unmyelinated. So that non-myelinating Schwann cell forms that indentation and then kind of surrounds and protects several unmyelinated axons. A myelinating Schwann cell is going to sheath only one axon. So each of these internodes that you see here, remember the internode is the area that's myelinated, each of these is actually one Schwann cell that has wrapped itself around that axon. So this here is one Schwann cell. So basically that Schwann cell just like flattens himself out really thin, and then he's going to fold himself around and around and around that axon in the center. And that's different than those oligodendrocytes that we saw, which remember the oligodendrocyte had its cell body, right? Here's an axon, here's an axon. Sorry, my poor drawing skills. And so he's going to have a process that comes out that has flattened itself, and it's going to wrap around here, and then he's going to have another process that comes out and it's going to wrap around here. You might have another that comes out and wraps around here to form our internodes. So that was the oligodendrocyte in the central nervous system. The Schwann cell in the peripheral nervous system is going to be, like I said, it's one cell that stretched himself out into a pancake, and then he wraps himself completely around that axon. So we're myelinating, but it's a bit of a different process than in the central nervous system. So we have many Schwann cells that sheath the entire axon when we're in the peripheral nervous system. And those gaps between the myelinated segments, again, are called the nodes. And here's just a better image from your textbook of how that one Schwann cell, so this brown or tan cell is our Schwann cell, and you can see it's kind of starting to stretch itself out, and it's going to stretch itself out and wrap and wrap and wrap and wrap around that axon in the center. And that's different than this oligodendrocyte where we had one cell body and it has numerous processes that are going to wrap around and around the axon. We can have disorders, different disorders that can affect the myelin and disrupt the myelination. This is called demyelination. Chronic exposure to some heavy metals like lead, arsenic, and mercury can lead to demyelination of axons. We can also have different diseases, infectious diseases, or autoimmune diseases that can cause demyelination. Diphtheria, which is a bacterial infection. That bacteria produces a toxin which can damage the myelin and destroy the Schwann cells, and it can actually lead to a fatal respiratory paralysis. This is one of the reasons we have a vaccine now that most children get during their toddler years to help prevent diphtheria. So it's not a common issue in the United States anymore. Some of you may have heard or know someone or may personally have multiple sclerosis or MS. This is an autoimmune disease. So remember, autoimmune is when the body does not recognize itself as self and the immune system starts attacking part of our own body. And MS affects the axons in the brain and spinal cord. So with this disease, you can end up with lots of different symptoms of different severities, ranging from loss of vision to problems with speech, problems with balance and coordination and movement. This disease can be progressive and affects more commonly women beginning 30 to 40 years of age. Guillain-Barré syndrome is another autoimmune disorder that leads to demyelination of the peripheral nervous system. And with this disease, you can have weakness and tingling all the way up to paralysis. Guillain-Barré syndrome can be triggered by viruses, but most people affected by this do luckily fully recover. So this luckily is a demyelination that you can recover from. Just another quick note about how our neurons respond to injuries. We have more repair possible in the peripheral nervous system than the central nervous system. In the peripheral nervous system, if we have an injury, we undergo something called Wallerian degeneration. So if a peripheral nerve is damaged, cut, or crushed, the axon that's distal to that injury is going to degenerate, and macrophages, remember those are our big eater cells, are going to come and clean up the debris. So we have, say we had a nerve in the peripheral nervous system and we had an injury here. Our macrophages would come and kind of eat up any of the injured area. Whoops, I didn't mean to erase that entire thing. They would eat up that injured area and debris to kind of clean it up. And the axon distal to that, so the axon distal to the axon distal to that injury is going to get cleaned up by those macrophages. Our Schwann cells, remember, those are the myelinating cells in the peripheral nervous system. Those Schwann cells can then proliferate and form a cord following the path of the original axon. And that cord that they form, the Schwann cells form, basically makes a path for that new axon to grow into as the neuron recovers. So in this image on your right hand side, whoops, you can see we had some damage to this nerve. Our macrophages come in and they are going to clean up that debris, and then the Schwann cell is going to proliferate and form kind of a cord here. And then the neuron, the axon of the neuron, can kind of follow that path that the Schwann cell made for it. And so with this type of healing, we may eventually reestablish normal synaptic contact. The Schwann cells may provide enough of a path that the axon can grow where it needs to go. But it is possible the axon could end up growing off in a new direction and we would not be able to regain normal function. And it's also possible that the gap that's formed after those macrophages clean up the injury is too large, or the ends are no longer aligned in the right orientation, the ends of the axon that have been damaged, that the Schwann cells are trying to reconnect. Surgery, though, is sometimes performed to fix this. So say we had a peripheral nerve injury and we had an injury here where the two ends of the axons are kind of not in the right orientation for those Schwann cells to form a cord. The Schwann cells would be, say, going in this direction. And so then our axon would end up growing this way and wouldn't be able to connect here. But surgeons could actually align the ends of the tissue so that it can reconnect. Unfortunately, in the central nervous system, nerve regeneration is more limited, and it is also more complicated because more axons are likely to be involved. Think about if you were in, say you had, say this is my very bad stick figure person. So say in our peripheral nervous system, we just have not just one or two, but we've got just a small number of nerves running through the peripheral nervous system, say within our arm. It's not completely packed with nervous tissue. We just have those peripheral nerves running through. So if we get an injury here, we might just be injuring 1 or 2 peripheral nerves, versus if we were in the brain, our brain is basically just jam-packed with all those neurons. And so if we have an injury come in here, we've got lots more neurons, lots more axons that are likely to be involved in an injury there because they're just packed in so tightly together. Our astrocytes are also going to limit the healing because they produce scar tissue, which can prevent that axon growth across the damaged area. So if we had a neuron that, had some damage to it, and we had this gap here, and then our astrocytes might produce scar tissue, and that prohibits that neuron from growing back together. The astrocytes also release chemicals that can block regrowth, unfortunately. And this is our last slide for this lecture. Just a thinking question about brain tumors. So remember when we talked about cancer, we talked about one of the ways cancer forms is when we have cells dividing by mitosis. If we have a mutation or abnormality in one of those cells, and it keeps rapidly dividing and dividing and dividing. So in our brains, our stem cells stop dividing after about age 4. Remember, this is one of those tissues where we don't continue to divide, we don't continue to produce neurons over and over. Those neurons, once we reach about age 4, we've got as many as we are going to have for our adult life. So if this is the case, if our neurons don't continue dividing in adulthood, how do we end up with brain tumors? Right? Because we would expect brain tumors to be coming from cells that are dividing and there's some sort of mutation. So if neurons aren't dividing, where do our brain tumors come from? So take a minute, pause this, and try and think. There are two different ways that in an adult we would see a brain tumor. So in an adult, remember, in our nervous tissue, we not only have neurons, but we also have those neuroglia cells. The neurons might not divide, but the neuroglia cells do continue to divide in adults. In children, we can have tumors involving the neurons because those neurons are still dividing. But in adults, most of the brain tumors we see are brain tumors related to those neuroglia. So you might have heard of tumors like a glioblastoma, and that would make sense now that you know that we have these neuroglial cells. The other way that we can get tumors within the brain, think about back to our lecture when we talked about cancer, is that remember we can have a primary tumor somewhere, such as breast cancer. We could have a primary tumor in the mammary tissue, and then we could have a secondary tumor or metastases form within the brain. So brain tumors could also be metastases from somewhere else within the body.